So good evening, everyone. Thank you for joining us for our first spring event of the InHealth Precision Medicine Society, a new student-led organization that aims to explore the benefits and challenges of integrating precision medicine into modern-day healthcare systems. Precision medicine is delivering the right treatment to the right patient at the right time. It's not a relatively new concept in human history, as you can see here by the quote from Hippocrates. But only recently have we begun to empirically elucidate the variations in disease response thanks to the advent of next-generation sequencing technologies. Throughout the academic year, we hope to set the groundwork of precision medicine following the framework outlined here on the slide. Previously, we've held a talk on biostatistics, and today we will be holding a talk that will primarily encompass the genome module. Through our events, we want to prepare the next generation of healthcare leaders through symposia, discussion forums, webinars, and more. We also hope to provide a platform for our members to voice their contributions to developments in this emerging frontier. Today, we are honored to be hosting Professor Danielle Gilkes, who will be presenting her work in developing technologies for understanding the role of hypoxia in metastatic breast cancer.
Okay, so before we get started on our talk, we want to take a few moments to share our new blog, the InHealth Chronicles. On our blog, we aim to publish short articles about recent developments in precision medicine, making these discoveries accessible to the wider audiences. So our first post is called the Bringing Precision Medicine into COVID-19 ICUs, and it has been featured by the Johns Hopkins InHealth Initiative, the Center for Clinical Global Health Education, and the JHU India Institute. So if you want to check out our blog, there's a QR code in the upper right-hand corner. And also, we're recruiting some members to help contribute to the blog. So if you want to contribute to the blog, be like an editor or fix the web design or stuff like that, you can fill out the application, and the QR code for that is on the bottom right hand corner.
Okay, and today we will be hosting Dr. Danielle Gilkes, who will be presenting a talk titled Developing Technologies to Decipher the Role of Hypoxia in Breast Cancer Metastasis. She's an assistant professor in the oncology department under the breast and ovarian cancer program at Johns Hopkins. Dr. Gilkes completed her postdoctoral work in the lab of Professor Semenza at Johns Hopkins, who was recently awarded the 2019 Nobel Prize in Physiology for his work in elucidating how cells sense hypoxic conditions. Since August 2015, Dr. Gilkes has her own lab, and her research has focused on the role of hypoxic microenvironments in regulating the extracellular matrix in breast cancer. Recently, her research group discovered that tumor cells exposed to low oxygen conditions have an advantage when it comes to invading and surviving in the bloodstream. Their recent findings hold tremendous clinical potential for improving health outcomes for breast cancer patients. All right, I think I'll switch things over to Dr. Gilkes to present. I think you're muted.
I'm sorry about that. When I'm trying to share the screen, it says host disabled participant screen sharing.
You should be able to do it now.
Okay, let me try.
Okay.
That looks better. Okay. So can you see my screen?
Yes, yeah.
Great. And you can hear me okay, hopefully?
Yeah.
Wonderful. So thank you very much for that kind introduction. Today, I'm going to talk to you about the role of hypoxia in breast cancer metastasis, and I'm going to show you some of the technologies that we're developing in our laboratory to sort of understand this role. So before I can talk to you guys about hypoxia, I have to tell you what hypoxia is. So to understand the pathophysiological state of hypoxia, we first have to understand how oxygen is regulated in normal physiology. So here, that's what I'm showing you in this picture. Maybe I can turn my pointer on. Laser pointer. So basically, the atmosphere contains about 21% oxygen, but the oxygen has to diffuse from the air that we're inhaling in the alveolar spaces of our lung and then diffuse into the bloodstream. Here, the red blood cells will carry oxygen to all the organs in our body. So the oxygen levels in the peripheral tissue can range anywhere between 3 and 7.5%. and it depends on the specific organ. So we consider hypoxia to occur when the oxygen levels drop below a normal physiological level of oxygen concentration for any given organ. So in different organs, that can be a different level. In tumors, which is what my lab is interested in, tumor hypoxia will result from multiple pathophysiological interactions. So you have abnormalities in tumor vessel structure. So that's these kind of crazy looking vessels here that we're showing in the cartoon. And this kind of alters their function. So this can lead to a decrease in oxygen delivery relative to normal tissue. But what you can also appreciate here is in this growing tumor, that's these cancer cells here, you have a ton of cell proliferation. So as you have this increase in cell proliferation, you also have an increase in oxygen consumption by the tumor tissue. So the net result is that you have this decrease in oxygen supply from these bad blood vessels, but you have an increase in oxygen demand. And so in solid tumors, the majority of solid tumors do develop regions of hypoxia. So what does that look like? So here I'm showing you a full cross section of a tumor. Now this tumor was derived in a mouse model, and I'm showing you some brown stain. This brown is stains to mark the hypoxic cells that are in this tumor. We can go and take a little bit of a closer look, and you can see right here where my pointer is that there's a blood vessel. And within a very short distance, really on the order of 200 microns from that blood vessel, you can see that the cells are already hypoxic. So that's the brown cells here. So I summarize this basically in a cartoon where you have a blood vessel that's in the center here. And then you have cells that are surrounding the blood vessel that are well oxygenated. And then at a certain distance from these well oxygenated cells, you have regions of hypoxia. And then even further from that, where we have absolutely no oxygen concentrations, you have regions of dead cells or necrosis, because the cells need some amount of oxygen to survive. So what kind of oxygen concentrations and how can we measure this? So here we've used an optical oxygen detection probe. And using this probe, we can measure, we attach the probe to a micrometer, and we can measure the oxygen concentrations in a live animal. So essentially, think about it as a needle-based probe that we've used a micrometer to deliver the needle to the center of the tumor here. And at that center point, the oxygen concentration is nearly zero. And this white tissue here, that's dead tissue within the tumor. So this is necrotic tissue. And then as we retract the needle, we can continue to measure with time oxygen concentrations. And here we're plotting this as a partial pressure of oxygen. The oxygen concentration here, if you can think that we're breathing in air at 21%, in the sort of normal tissue out here, you're reaching oxygen concentrations of about 8%. So this is just to illustrate tumor hypoxia. Okay, so now I'm showing you a mouse, but why does this really matter? So there have been some groundbreaking clinical studies in which oxygen measurements have been made in breast cancers in patients. So here, in this case, the measurements were made in situ by using an Eppendorf microelectrode. And I'm showing you just one of the studies here. So in this study, there were measurements made at over 1,000 points within normal breast tissue. And 16 participants were a part of this study. And you can see that the median partial pressure of oxygen for these patients is about 8%. And I'm just doing the conversion for you because we're in partial pressure here. Whereas if we look at breast cancer patients, you can see a huge shift in the oxygen concentrations. And here, these patients have on the order of about 1.5% oxygen in their tumors. So this is a study in breast, but remarkably similar results have been obtained in meta-analysis of studies involving over 700 patients with cervical cancer, 500 patients with head and neck cancer. And in many of these cancers, the studies have gone on to demonstrate an association between partial pressures of oxygen of less than 1% with decreased disease-free survival. So that's why we think that this is an important clinical question to address. So these patients that have much lower oxygen concentrations in their tumor are more likely to suffer a relapse. So what happens under these low oxygen concentrations? I mean, I showed you the necrotic core of the tumor. So why aren't all these cells dying? They should be. How do cells adapt to this low oxygen concentration? So as Mahmoud mentioned in the beginning of the talk, HIF-1 alpha was discovered here at Hopkins in Greg Simmons' lab, where he later got the Nobel Prize for this discovery. And he showed that, that metazoans have a complex and well-orchestrated cellular oxygen sensing pathway. And among these pathways, one of the very important factors is hypoxia-inducible factors, or HIFs, and they've established central roles in oxygen sensing. And I mentioned HIF-1 was first purified and cloned here at Hopkins. The Hopkins lab purified the protein HIF-1 alpha, and later HIF-2 alpha was discovered. And they have a certain degree of similarity between the two proteins. What you'll notice is that the half maximal induction of HIF-1 alpha occurs at about 1% oxygen conditions. But the HIF-1 alpha binding protein, HIF-1 beta, really doesn't sense or respond to these decreased oxygen concentrations. So you can think of HIF-1 alpha as only being induced under low oxygen concentrations, but HIF-1 beta sort of hanging around. But the two puzzle pieces need to come together to induce transcription of target genes that are important to adapting to this hypoxic concentration. So how exactly does this happen? So here I'm showing you HIF alpha. So think about it as HIF-1 or HIF-2, whichever your favorite HIF alpha target is. So when there's oxygen present, the oxygen leads to oxygen-dependent hydroxylation. And that occurs by these PHD proteins. Then what this does, these oxygen residues or these OH residues that are on HIF-1 alpha allows a protein called VHL, which is an E3 ubiquitin ligase, to notice HIF-1 alpha. So only when it's hydroxylated in the presence of oxygen can VHL notice the protein. And when it notices HIF-1 alpha, it ubiquitinates HIF-1 alpha. And that sends a signal to send it to the trash can and basically destroy the HIF-1 alpha protein. So when oxygen is present, we don't have a lot of HIF-1 alpha available. But as soon as oxygen is limiting, now HIF-1 alpha is no longer hydroxylated. That means VHL can't find it in the cell anymore. So it's protected. HIF-1 alpha can then bind to the constitutively activated HIF-1 beta protein that I mentioned earlier. And together they can go translocate to the nucleus, bind to target genes. And many of these target genes are important in cancer. And more specifically, we're finding they're important for metastasis, including angiogenesis and metabolic reprogramming, the stem cell-like phenotype, many of these things. So my lab is very interested in studying this phenomenon and to understand what role HIF-1 alpha plays in the metastatic process. So one of the things that we've done is we recently, as you guys were mentioning the genome, so we recently did a large-scale genomic analysis. We essentially exposed 32 different breast cancer cell lines to either 20% oxygen levels. So that's the air that we're breathing, and that's what our cell culture incubators have that we incubate our cells in. And then we took a duplicate set of cells and exposed them to hypoxic conditions, in this case, 1% oxygen conditions. So under 20%, you have low levels of HIF. Under 1%, you have high levels of HIF. And then we asked what genes are upregulated the same way in the 32 different cell lines that we tested. And what we were really surprised to find is that in each individual cell line, there may be 3,000 genes that are changed in their expression, but only 42 genes that we found were similar amongst the cell lines. So that's a little bit scary in some ways, but we did find this 42 gene set. And we went on to look at patients with breast cancer and to stratify those patients. So this is a Kaplan-Meier curve. So at the beginning of the study, at 0 months, all of the patients are alive. So we have overall survival of 100%. And then we separate out the patients with low expression of this gene signature or with high expression. And we follow these patients over time and we censor them at different time points. So this is showing how many patients, so 80% of the patients with high hypoxic scores are alive at 50 months or close to 50 months after their diagnosis, whereas a higher level if they have a lower hypoxia signature. So you can see that sort of out here where the two different patient populations really segregate between low and high. So we think that based on this study, and of course, many other studies in the literature, that HIF-1 alpha is prognostic and is important for patients and for predicting patient response. This I'm just showing how if you do a search for hypoxia and metastasis, sort of the number of publications. So really you can see in the early, even in the early and late 1990s, there was really not very much going on in the field of hypoxia and metastasis. And then I'd say right around 2009, things started really upticking. And overall, if you do this search, hopefully you'll find about 4,500 papers. This slide's a little bit outdated, so probably more. So it's really just to show you that more folks are interested in studying the role of hypoxia and metastasis. So what is metastasis? Metastasis is basically the spread of cancer cells from the primary site to distant sites. And so here we are highlighting in this review different proteins that are hypoxia-inducible factor 1 dependent, and that are also involved in the metastatic cascade. So here, this is supposed to be a cartoon of a primary tumor, and cells must be able to, or cancer cells, in order to metastasize, must be able to break through the basement membrane that's surrounding them. And then they have to successfully intravasate. We call this a process of intravasation, where the cancer cell needs to squeeze into the blood vessel. So the blood vessel is covered with these endothelial cells, and somehow the cancer cell needs to push through. Now you can imagine a cancer cell in the bloodstream. Usually it's sitting still. The cancer cell here in the bloodstream has to survive all of these shear stresses. They're essentially being asked to swim, but they were never really supposed to. So you can imagine the cell sort of swimming in the bloodstream. Now it has to actually grab on to the blood vessel wall again in order to get out of the bloodstream. And so that's a picture of extravasation shown here. And then now the cell is found in distant soil that it's not used to being in. So this breast cancer cell has now found itself in the lung or the brain or the bone or the lymph node, and it now has to figure out how to survive. So luckily for us, this is a very inefficient process, which is great because that means metastasis doesn't occur, is not a major, doesn't occur to everyone, but it's important because that is what kills patients with solid tumors. So here I'm just showing you that HIF-1 alpha can regulate a lot of proteins that are involved and that help the cell to metastasize. But we sort of have this slight paradox, right? So in the beginning, I showed you that under hypoxic conditions, HIF levels are very high. But once oxygen levels increase, HIF proteins are very quickly degraded. So this led my lab to ask the question, how reversible is this hypoxic response? So if you have a cell that becomes hypoxic, but then it moves to a more oxygenated region of the tumor, or it enters the bloodstream, which I told you at the beginning is very oxygen rich because that's what's delivering oxygen to all of our cells, it sort of leads us to the question, what happens? Is there something memorized? So the first thing that my lab did when we opened was we wanted to address the question, can cells that experience hypoxia escape and really metastasize, or will they fall into that necrotic core that I showed you earlier? So how did we do this? Well, in the field right now, the only way that we can monitor hypoxia is to mark cells that are currently hypoxic. So we needed to figure out a method that we could follow these hypoxic cells and then monitor them as they sort of made it to the bloodstream. And that was what the cartoon was that I just showed you. So we came up with this approach. So we've designed a plasmid. So it's essentially just think about a piece of DNA that we can deliver to a cell. And this is all in the laboratory setting. So we've delivered this certain plasmid to the cell. And the because we've delivered this plasmid or piece of DNA, the cells express a red fluorescent protein. So they're all red. They're very cool. You can look at them under the microscope and all of the cells are red. Then we delivered a second plasmid or piece of DNA to the same cell. This plasmid contains these hypoxia response elements. And that's where HIF binds to these hypoxia response elements. But then it drives the expression of a gene, which is known as Cre recombinase down here. And you can think of Cre recombinase as a pair of scissors. So under low oxygen concentrations, under high oxygen concentrations in the air, there are no HIFs around. So these scissors are not being made. This Cre recombinase, which acts as a scissor. However, under low oxygen concentrations, when we have HIF-1 alpha and HIF-2 alpha expressed at very high levels, they'll bind to these hypoxia response elements, and they'll drive Cre expression. And Cre acts as scissors, and it basically cuts out these LOX-P sites here shown in purple that surround this RFP protein. So once this RFP protein is cleaved out, now the cell expresses GFP. So does our system work? So hopefully I'll convince you that it does. So here we've exposed cells to 20% oxygen and they are red. We've exposed them to 5% oxygen and they still maintain a red color. And this is important because we know that normal breast tissue is close to around 5 to 8% oxygen. So we don't want our switch to happen until the cells are exposed to hypoxia. And then under low oxygen concentrations, you can see the cells are green. Now, hopefully, I will be able to play this movie. Let me see. So here we have in the lab a microscope, which we can follow cells over time. So at time 0, we've exposed the cells to hypoxia. So that was when they were all red. Let's see if I can make it play again. Maybe not. Well, at time 0, we had, I can't get it to replay again, but that's okay. At time 0, we saw all the cells were red. And then as we exposed the cells and monitored them over time, you see that they changed to this green color. Now, what's important is that the green color is permanent. So even when the cells leave the hypoxic region, we're still going to be able to find them. So think about if all of us in this room had a red t-shirt on, and then we were all asked to maybe go over a tightrope, walk a tightrope. And if we were successful at doing that, so we were successful at surviving this hypoxia insult, then we were given a green t-shirt to change into. And because of that, now we're able to find all of these successful cells in a tumor and at metastatic sites. So I'll show you what that looks like. Of course, now it wants to play again. Okay, so what we can do also is we can generate sort of tumors ex vivo. So here I'm showing you that we can take cancer cells and then we can put them in a certain media that makes them not want to sit down, and we can form these sort of mini tumors. So what does that look like? So in this case, we're using MCF7 cells. These are just breast cancer cell lines that we can grow in the lab. And you can see at time 0, they are all red and they're sitting here in this nice collagen matrix. And then over time, you can see the existence of these green cells starting to form. You can do this in metastatic breast cancer cells. So in this case, these are human metastatic breast cancer cells, MDAMB231 cells. Again, they start red, and then you can see that they're highly invasive because they are metastatic breast cancer cells. And you can see that the cells also turn green with time. So they're becoming hypoxic even in our cell culture. Here is what this looks like in a 3D. So we're kind of moving into the center, and you can see that these GFP positive cells can leave the center of this 3D modeled ex vivo tumor and actually migrate to the very edge of the region. We can also use the optical probe that I showed you last time to ensure that our cells are really expressing hypoxia. So in this case, we have number 1, which is measuring the very center of the spheroid that I showed you. And number 2, the probe is measuring far away, so in the matrix, not not near the cancer cells themselves. So again, you can see that there's very low oxygen concentrations within these 3D sort of organoids or spheroids or tumors. Okay, but what I mentioned to you in the beginning is that I'm curious about metastasis. So we use, one of the models that we use in our lab is a murine model of metastasis, wherein we take metastatic breast cancer cells and we implant them into the memory fat pad of a mouse. And then we allow the tumor cells to grow in that mouse. And then we can either remove the primary tumor, as one would remove a tumor from a patient, and then, or not, and I'll show you both scenarios. And then we can measure lymphatic metastasis, circulating tumor cells, lung metastasis, brain metastasis in these mice. And the question that we're asking is whether these GFP positive cells can actually metastasize. And do they do a better job at it or not? So here's what the tumors look like. So at the early time point here, when the tumor is much less than 2 millimeters in the mouse, you can see that the whole tumor is red. So there are no real hypoxic cells. And then as you look with time, you can see, first of all, just as I showed you at the beginning, that the hypoxic cells really line up against this necrotic core. And that's all this black tissue here is basically dead tissue, this dead tumor tissue here. But then you can see with time that the tumor becomes very hypoxic. There are a lot of green cells, and the green cells can actually migrate into the regions that have higher oxygen concentrations. So we're showing here that our reporter construct that I showed you at the beginning is really working. And then we've moved on to investigate whether these hypoxic cells can metastasize. So how we did this study is, again, we took our breast cancer cells, we implanted them into the mouse memory fat pad. We excised the tumor at day 20. So we performed a small surgery to remove the tumors, and then we followed them over time. So the primary tumor at the time of removal only contained 20% GFP positive cells. The majority of the cells were DS red because not all of the cells were hypoxic. But then if we look at the lungs, when we harvest them, these are the lungs from the mice, and this is just a whole mount image. So we can essentially just take the lungs out of the mouse, put them on our microscope, and look at green versus red staining. And we can also section, make very thin sections of the lung. And hopefully you can see that this whole thing here is one of the lung lobes, one of the 4 lobes of lung in the mouse. And you can appreciate the green cells are the hypoxia, the cells that were exposed to hypoxia in the primary tumor, whereas the red cells had enough oxygen concentration. So what can we measure this? We use different techniques to measure this. And what I can tell you is that although the tumor at removal only contained 20% of cells, more than 45% of the cells in the lung were GFP positive. So if we look at this ratio, we can get an idea that a GFP positive cell or hypoxia-exposed cell has a 5 times increase in its ability to form a metastasis. If we look at, this is a lung section, and here we see that there's about a 50-fold increase in the ability of a cancer cell that's been exposed to hypoxia in the primary tumor to form a liver metastasis. Lastly, we looked at the brain. And in our model, the cells don't readily metastasize to the brain. And we can save that we can talk about the blood-brain barrier another time. But what we do find is that the cells that we do identify, the single cells or multi-cell structure in the brain are mostly GFP positive. We can also play some cool, we can use some very cool novel ways to perform imaging. So here I'm showing you a whole mount of a brain in the mouse, and you can hopefully appreciate this green section of tumor cells. What we can do, because we have so few metastases in these brains, is we can perform what's known as tissue site imaging. So in our mouse, using this technology, you can think about it as taking a section of tissue and putting it on a conveyor belt. And then at the end of the conveyor belt, there's a microscope. So you're sectioning a piece of tissue, putting it under the conveyor belt, imaging, sectioning the next piece. And if you continue to do that, you can reconstruct the entire brain of the mouse. And so, oh, okay, I'm sorry. I thought I had a movie here. I guess I don't. But this allows us to really quantify the number of GFP positive cells in the mouse, in the mouse brain. And there are some other cool techniques that you can do that we've borrowed from neuroscientists where there's actually a brain atlas from a mouse, and you can overlay your pictures onto that to figure out exactly where the cancer cells went in the brain. So there is some cool technology there that maybe we can discuss at a different time. We also generated mice that express this reporter. So I told you we took this little piece of DNA and we put it into cells. And then we took those cells that had those DNA constructs and we put that into the memory fat pad of the mouse. In this case, we're generating a mouse that in every tissue in the mouse has this oxygen sensor. And we cross it with a mouse that is really cool because he is totally red. So it's similar to the cell line. So if you saw the mouse like on my desk, if you had a filter, so some sunglasses with the appropriate filter and a light, in this case, a light that would excite the protein, you can actually see that the whole mouse is red. And then we have this oxygen responsive construct. So in the cells that have low oxygen concentration, you're going to see GFP signal. What we had to do, though, is we had to cross these mice with mice that develop spontaneous breast tumors. And I won't go into all the genetics behind how that happens, but we essentially had to take 3 mice and cross them together so that we could have a mouse that forms spontaneous tumors that are red, that will change to green when they experience hypoxia intratumorally. And so what does that look like? Well, the model is very nice because it really sort of mimics breast cancer pretty well. So in this case, you're seeing fat cells. Those are these big, wide vacuoles that you see here. And here, the purple cells are hyperplasia. So we're starting to see abnormal growth in the memory fat pad of these mice. Then if we wait a little while, we see these DCIS-like lesions. So these are lesions that haven't invaded into the surrounding fat tissue, but you can see that they're forming and they're occluding a lot of the fat. Later on in time, they develop early carcinoma where they've essentially now the cancer cells are invading all of the normal tissue, and they move on to very invasive carcinoma, and actually they do metastasize. So this is what the histology of the mouse tumor looks like. But here I'm showing you the red and green color. So here in the hyperplasia stage, we don't see any GFP positive cells. And in the DCIS lesions, you really have to zoom in to see hypoxia occurring in the lesions. And then by early and invasive stages of carcinoma, you can see that we have a lot of green cells and fewer red cells. This is just looking in more detail at the DCIS lesions where you can sort of see the emergence of hypoxia occurring. So basically, we have a mouse model now in the lab that we can study hypoxia both from a developmental standpoint, so we can look at organs and how they develop, how mice would develop in utero, because we know that they are under hypoxic conditions, but we can also study metastasis. So here we can remove some of these organoids from the mice and again just grow them in culture. And you can see that even the organoids from these mice, when we put them in cell culture, you can see them developing this GFP positive population with time. So they're becoming hypoxic. I don't know, this may or may not be a movie. Yeah, so this is just monitoring the growth of these organoids in a cell culture dish. So you can see with time, you can take so these are organoids, so these are tumor cells taken out of the mouse and placed when they're at a very early lesion stage into a collagen matrix, which we can then image with our microscope to look at the presence and the induction of hypoxia. But what was more important was to look into our transgenic mice. And in this case, these mice have metastasis to the lung. So in the transgenic model here, we're looking at the lungs of the mice and we're imaging GFP positive and DS-RED positive lesions, and we're counting them. And what we find again is that even in this mouse model of metastasis, we do see an increase in the ability for the GFP positive cells to form a metastases. Then you can perform a secondary model where you take tumor from the primary model and put it into recipient mice and ask the same question. And again, we see an increase in metastasis from the GFP positive cells. Okay. So in the interest of time, because I want to make sure that we have plenty of time for some question and answers, I'll tell you that we decided to look at each step in the metastatic cascade to figure out where these guys are becoming more powerful. In the essence of time, I'm just going to tell you that we did not see a dramatic difference in invasion in these sort of early steps. So we asked whether the cells were more primed to survive the stress in the bloodstream. So we didn't see really a difference in this stage. So the next question we asked is, well, are they better at getting into the bloodstream and or surviving this process of having to live in this sheer stress environment? So this is just looking at local invasion. So I'll go past that and we'll jump right to the circulating tumor cells. So what we did was we asked if the cells in circulation, if we had more GFP positive or more DS-red positive cells. And what we found is that we actually had more GFP positive circulating tumor cells. So we thought, you know, they're not doing too much with invasion. Are they better than at getting in or actually at surviving? So in order to test that, we looked at the number of apoptotic circulating tumor cells. So apoptotic means dead cells. So what we found is that there were many more dead DS-red or red cells than there were green. So this suggested to us that the GFP positive cells are somehow more primed to survive in the harsh conditions of the bloodstream. And so that's what we really focused on. And then we did not see differences in the ability for the cells to get out of the bloodstream and or to form colonies once they got out. So next, we wanted to understand why. Why are they better? What's happening in the bloodstream, and what powerful tools do the GFP cells have? So what we did was we went back to the literature to say what happens in the bloodstream. What are the major stressors besides the shear stress? And we looked and found that many recent reports have suggested that oxidative stress or reactive oxygen species is a limiting step in the metastatic process because it harms the circulating tumor cells and it causes them to die. So we questioned whether the GFP positive cells were more resistant to this oxidative stress. And so in order to do that, we had to measure the reactive oxygen species directly. So we isolated GFP positive or RFP positive cells, cancer cells, directly from the blood of these mice. And we did a staining procedure to assess their level of ROS. And in order to mitigate ROS, we used this compound called NAC. So we sorted the cells and either treated them with this, this way to prevent reactive oxygen species or not. And basically what we found is that the GFP positive cells had much less ROS than the DS-red cells. So this suggested that the cells were protected from the reactive oxygen species. However, if we treated either the DS-red cells or the GFP positive cells with this compound that reduces the levels of ROS, we reduced the level of the DS-red cells down to the GFP positive cells. So now it's a little bit hard to kind of follow, but just think about the bloodstream as having a lot of stress, and that the hypoxia-exposed cells are somehow able to deal with this stress. And this is another assay that we measured this in. So, but I'm gonna move through that and sort of go at what type of tools you can use to figure out, to kind of address these questions. So we wanted to know what causes this ROS-resistant phenotype. So in order to do that, we use the technologies that you guys were alluding to before, some more genome technologies. So we removed the primary tumors from the mice at an early time point. We sorted them based on their color to red and green. So we used flow sorting to to separate the two different populations. And then we waited 45 days and we did the same thing in the lung, and we sorted the red and the green cells. Then we were able to perform RNA sequencing on the different populations, the red, the green, and the primary tumor or the lung. And so here we found that 164 genes were upregulated in the green cells compared to the red cells, while 43 genes were downregulated in the GFP cells compared to the red cells. In a secondary experiment, we said, well, hey, everybody in the field of hypoxia basically just exposes their cells to 20% oxygen or mimics hypoxia by exposing the cells to 1% oxygen. So let's do the same thing and see if what's happening in vivo in our mouse is the same thing that's happening when we play out this experiment in vitro and in the laboratory-based setting. And it was a little disheartening again to see that the in vivo situation, when we measure the hypoxic cells, the gene signatures are quite different than when we take a cell, the same exact cell, and just play with them in the lab, expose them to hypoxia. So we really only had a 41 gene set that had a consensus between our contrived lab-based experiment versus the actual real deal in the mouse. And we looked at the different gene sets and we asked which one could be most prognostic for breast cancer patients based on the different gene sets that we have here. And I'll suffice it to say that the intratumoral hypoxic gene signature, that is this 164 genes, if we stratify patients based on whether or not they have this gene signature, we could predict distant metastasis-free survival. Likewise, the 41 overlapping signature did a very good job too, stratifying the patients. So those that had a very high hypoxic signature had a worse overall prognosis. Oops. Sorry about that. I want to go back. I think suffice it to say that the 798 signature did not. I think I've sped through that particular survival curve. Sorry about that. So the summary so far, and I may have to leave it here so that we can have a little bit of a discussion, is that hypoxia occurs in tumors due to an imbalance between oxygen supply and oxygen demand. That's what I told you in the beginning. I told you that hypoxia promotes HIF protein expression, and that leads to aberrant gene expression patterns. And we looked at those gene expression patterns by doing RNA sequencing. We developed this hypoxia fate mapping strategy by delivering by delivering a piece of DNA to either cancer cells or to a whole mouse. And by doing that, and by having this permanent hypoxia marking strategy, we could tell that hypoxic cells are 5 times more likely to form a lung metastasis than cells that have never experienced hypoxia. And I think that's what we published on most recently, but what we're now really delving into is trying to understand why they seem to be even more potent in the liver and the brain. And so that leads on to my next summary statement, is that hypoxic cells seem to have a preference for the liver and brain over the lung. But why is that? What could be causing that? We also showed that cells that are exposed to hypoxia are poised to tolerate and survive the oxidative stress that they encounter once they enter the bloodstream. And we showed that tracking cells that become hypoxic en route to metastasis reveal that there is a certain gene signature that is protective against ROS. But what is the mechanism? So I'm not going to go into the mechanism due to kind of, well, I'll touch on it. So we think that based on our RNA sequencing results, you're able to, from your RNA sequencing results, you can look at pathways that are enriched. So not just individual gene signatures, but actually whole pathways. And this one pathway is called the NF-kappa B pathway. And we found it to be highly enriched in our GFP cells, both from the primary tumor and from the lung. So we did another fancy experiment where we can, again, using a fluorescent color, identify cells that have high expression of this NF-kappa B pathway. And it turns out that the GFP positive cells do have an increase in NF-kappa B signaling. And that's both in the primary tumor, the blood, as well as in the lung. And what does that look like? Here's our lung. Here's a portion of the lung that has GFP positive expressing cells, and that overlays with this purple color. And the purple color are the cells that are expressing high levels of NF-kappa B. And then we have the DS red cells, and we do not see NF-kappa B expression there. So we have gone on to look at downstream targets that could prevent cells from this ROS insult, but I'm not going to do a deep dive into that here. So I'm just going to close out here because I think I've given a lot of information. Hopefully I haven't confused you guys too much. And I'll thank the folks in my lab, the graduate students pictured here, as well as our collaborators, both within Hopkins, as well as at other institutes like the FDA and Ohio State University. And so with that, I'll stop sharing my slideshow and entertain any questions that you guys might have about the system or why we did certain things the way we did.
Okay, thank you so much. That was an excellent presentation. Thank you. So I think we'll switch over now to the Q&A session.
Did I lose you all, or did you guys kind of understand the color marking strategy that these guys started out a certain color, and then when they felt the hypoxic stress, they were able to change color?
A lot of that actually was very relevant. I mean, at least for, because I'm taking a developmental bio class, so a lot of the stuff regarding LOXP sites and CRE was very familiar.
Okay, good. It's hard to know, if you haven't taken those classes before, sometimes it can be very difficult to try to understand. So try to do the scissor analogy where you have a way to kind of cleave something out and then turn something else on. And so that's not a strategy that our lab generated. It's just we borrowed it. We generated that second vector that drives CRE under hypoxia. That was kind of the more novel thing that we did versus that the first vector is, just has those CRE-lock sites in it.
Yeah, so I think if anyone wants to raise their hand, and we'll just start fielding questions. Okay, Xinwei, go ahead.
Hi, Dr. Gilks. I don't know if I pronounced that correctly. Just a super general question. I know, you mentioned that hypoxic cells seem to have a preference for liver and brain over lung. Do you think it has anything to do with cancer cell metabolism, or do you have any idea how we can interpret that?
I mean, I think that's an excellent question. That's definitely something that we're looking into right now. So we finally, one difficulty, especially with the brain, I showed you that there are very few cells that metastasize to the brain. So we could not collect enough of those cells to kind of perform assays to look at if their metabolism was changing or what. So we've turned to a new technology. It's called spatial transcriptomics. I'm not sure.
Oh, yeah.
Have you heard of it?
I've only heard of it because they generate fancy graphics, and I like graphs.
Yeah, so it's very cool, because what you can essentially do is you can have an image of your tumor, and you can drill into each 50 micron spot and ask what gene expression patterns are occurring in that spot versus what's occurring in the spot just next to it or the spot just next to that. So we have results back that we've been able to apply that sequencing platform to, and we are trying to get at your question right now. The other thing that folks have asked us about is whether or not, because the liver has a lower oxygen concentration and the brain has a lower oxygen concentration as compared to the lung, are the cells more prepared to be in a lower oxygen dependent environment? And I can't answer that yet. But that's an alternate hypothesis that folks have asked us.
Thank you.
Thank you for that question.
All right, does anyone else have a question? Go ahead, Jojo.
Hi, Dr. Gilks. Thank you so much for that talk. I just have a quick question about the slide where you showed the overlap between the gene signatures from the intratumoral study samples versus the in vitro ones.
Mhm.
So I was just kind of wondering, what do you think are some of the reasons why those two have a very low overlap? Does that just mean that a lot of the genes that you identified are less significant somehow, or does it point to something else?
So that is an excellent question. So that's something that we've been really trying to address, because we want to make sure that whatever we're doing in the lab is really mimicking, what we're finding in the tumor. So one way that we've addressed that is we've exposed cells. One thing that we've thought about is that, in the primary tumor, these cells are exposed to hypoxia for longer than a day. Like these cells are sitting in the zone for a long time. So we've gone on to look at cells that we call them chronically exposed to hypoxia. So for 5 days or for 10 days, and that signature matches a lot better to the intratumoral. So we think that could be a timing thing, how much time the cells are there. The other thing that we think is that in our lab, we expose the cells to 21% oxygen, to air. Nowhere in our body are we exposed to 21% oxygen, even when we're breathing, maybe in our mouth. But then as soon as we breathe in air into our mouth and it goes into our lungs, the partial pressure of oxygen is already dropping. So even in our lung, we're not even at that level. So it may be that we're comparing apples to oranges in the lab. So we're comparing 21%, this kind of super physiological level of oxygen, to 1%, versus when we're in the tumor, we're really probably comparing 4 or 5% oxygen to maybe 0.5% oxygen, So it could be that the gradient of oxygen is different also. So I think that's an excellent question, because we need to know that answer if we're going to trust the results that we're doing in the lab, and we need to know which one's correct.
Right.
And the spatial transcriptomics is helping us a lot with that too, because with that technology, we can actually say, okay, we know that near the necrotic core, the cancer cells are really, really hypoxic. I mean, they're almost dead. They're almost falling into. So what is the difference between those cells that kind of fell into the swamp versus those cells that are farther, more into the oxygenated region? So I think we're borrowing from this kind of newer technologies, we're able to get at some of those questions a little bit better.
Got it. Thanks so much.
Yeah, thank you for that question. It was a great one.
All right. Does anyone have another question? Feel free to raise your hand. I know I had a question regarding just like the clinical implications of these findings. Like, how do you see this impacting care? Do you think that, this could be a way, this can be, for instance, leveraged to develop certain therapies that will, make it less likely for patients to move, from stage 1 to breast cancer to metastatic cancer?
Definitely, that's an excellent question. So in studying the NF-kappa B pathway, we think that we found specific genes that are protective, that are involved in protecting these cells while they're in the bloodstream, sort of kind of giving them a shield to allow them to metastasize. So right now we're using CRISPR-based approaches to say, okay, we believe that from our RNA-seq study, we believe that say gene X is really important to protect the cell when it's in the bloodstream and allow it to metastasize. So now we'll use CRISPR technology, and we will knock out that specific gene and ask if it's important. And moreover, we've developed a CRISPR technology technique to only knock out a certain gene in cells that are hypoxic, but protect the cells that are not hypoxic. So we can really kind of delve into what exactly the role of hypoxia is. Because clearly we have DS, even though we're saying they're 5 times more likely to metastasize, we still got to worry about all those DS red cells too, right? So we have to we can't think that hypoxia is the end all be all, but certainly I think it plays a role, and maybe it's letting things out of the gate much faster than they would be. So I think that's important. Right now, to be honest with you, many people have developed gene signatures for hypoxia. None of these are used as biomarkers in the clinic to prognosticate what a patient, to give them any indication of how, deadly their tumor might be. And we don't have treatments available right now that would make coming up with biomarkers and testing those biomarkers needed. So it's sort of this, it'd be great, somebody would like to know, how serious is my tumor? But if you don't have a product to give them, to fight that hypoxia, then, how important is that finding? So we really have to figure out, and targeting HIF-1 alpha has been tried, and there are several ways that you can target HIF-1 alpha, but there are a lot of off-target effects. There is a very nice HIF-2 alpha inhibitor. It's specific for HIF-2 and not HIF-1. I forget who bought, the company was called Peloton, but somebody just bought them out. One of the big companies bought them out. And that's going to be used really in the renal cell carcinoma space, where HIF-2 alpha plays a predominant role. But we haven't tried that here because we really think we need to attack both HIF-1 and HIF-2 because they're both playing a role. So hopefully, it gives us hope that this HIF-2 alpha inhibitor was developed. So it means you can target a transcription factor. So now let's go do it,
Yeah. Okay, thank you so much. Did anyone else have any other questions? Okay, Shinwei, yeah.
Sorry, just a final quick question. I know your research primarily focuses on breast cancer, but like, what is special about breast cancer metastasis that makes it like different in hypoxic settings?
So I don't think that that's necessarily true. I think some of the things that we're finding here should very well be tested in other solid tumor cases. I'd say the reason why our lab has a focus on breast cancer, besides being in the women's malignancy program, is that a lot of times developing these models are very difficult. So for example, we know very well how to deal with these mouse models of breast cancer metastasis. But if we move to prostate, we would have to figure out all of those models sort of starting from square one. Sometimes we're limited just by what our focus has been, but certainly I expect these findings to be very relevant in other cancer types, solid cancer types. But we'll have to get, hopefully we can get our prostate cancer guys to test out some of the things that we're finding here for us.
Thank you.
Yeah.
All right. Did anyone else have any other questions? Okay. We'll move on to, before we close this event, just wanted to give everyone a heads up of some exciting new events you can look forward to. So we're especially excited to be hosting Dr. Papadopoulos, a world-renowned geneticist and professor, to come chat with us about his work in developing a non-invasive liquid biopsy for early detection of cancer during the last week of April. We don't have an exact date pinned down yet, but check your inboxes for updates on this upcoming event.
And before we end our event, we'd like to let you know about a new internship opportunity for undergraduates. So the InHealth Initiative is seeking a business and marketing assistant for the summer of 2022. This is a really exciting chance to collaborate with clinical and IT teams across the Hopkins Medicine Network, including the Technology Innovation Center, the Institute for Clinical and Translational Research, and Precision Medicine Analytics Platform customers. We'll also be working to expand the precision medicine portal where all the InHealth resources can be accessed. So if you are interested in communications, digital content creation, marketing, and precision medicine, this could be a great opportunity for you. So to apply, you can scan the QR code for the job posting. And if you have questions, you can contact Sharon at InHealth for more information. And finally, we want to extend another huge thank you again to Dr. Gilks for taking the time to present this wonderful talk and answer all of our questions. So thank you so much, Dr. Gilks, and thank you to everyone here for attending our event tonight.